The deep sea is a challenging environment. Research in deep sea habitats is critical to resource management and the understanding of life on this planet. Deep Search is a partnership between the Bureau of Ocean Energy Management, the United States Geological Survey, and the National Oceanic and Atmospheric Administration Office of Ocean Exploration and Research. The Deep Search study was conducted between 2017 and 2022 in the offshore habitats between Virginia and Georgia. Through this study, our understanding of the coral, canyon, and cold seep habitats of the region was greatly expanded. Deep Search was comprised of researchers from government agencies along with academic professionals from leading universities under the lead contractors from TDI Brooks International. Together, they have explored more than 31,000 square kilometers off the coast of the southern United States. The Deep Search team set sail on the RV Atlantis in 2018 with the Alvin submersible on board to continue the study already begun on the deepwater habitats off the U.S. mid- and South Atlantic coast. This 15-day expedition featured daily dives in the Alvin, as well as nighttime operations such as mapping, core sampling, and water sampling.
My name is Mike Rasser, and I'm a marine ecologist with the Bureau of Ocean Energy Management. BOEM's mission is to responsibly carry out the management of the U.S. Outer Continental Shelf. The purpose of the project is to learn more about the habitats, canyon, seep, and coral habitats in deep waters off the U.S. Atlantic.
I mean, the overall goal of the project is to see what's out there, to find sensitive habitats and protect them. So the kinds of sensitive habitats that BOEM wants to protect are in 3 different categories. The canyons, the seeps, and the corals, all sensitive for different reasons. In the northern part of the study area are a bunch of canyons. There are corals on the steep walls of the canyons. There's a ton of productivity in the area because you get so much nutrient upwelling that happens here that it leads to high productivity, and you've got, whales and fish aggregations and everything lying above them. And then we have methane seeps. They're right at the heads of the canyons where you get lots of material being buried and degrading and turning into methane that's all bubbling up to the surface.
These are depositional environments that had really high organic inputs, a long time ago, tens of thousands of years ago. And as that organic carbon has been cooked and ruminated in the sediments, it produced methane. And that methane is now seeping out of the seabed and fueling all of these organisms that can feast from methane.
And then we get into some of the coral banks down here, and that's where we're going to get the coral reefs starting to form, but also really diverse communities of octocoral sea fans and lots of fish attracted to them.
When you think about the system as an evolutionary continuum, the microbial mats are the beginning of what is ultimately a deepwater coral hardground. That's what I'm interested in, is getting sort of a big holistic view of how these systems work.
I'm really excited to get my first look at this canyon. This will be the first time that we really get our eyes on these habitats. We're going to start up towards the head of the canyon and climb one of the walls. We'll be taking a lot of samples as we go. Lots of different samples of corals to look at the coral diversity, the animals living alongside them, thinking a lot about sort of the processes in the canyon where the food's coming down.
The really cool thing that I find fascinating about the seep environments is that we have ecosystems that are completely independent of the sun, which is rare on Earth. The sun fuels most life, but this is an exception to that rule. So everything that we can learn about these environments is relatively new to science, and it's exciting.
Today we're diving on the Blake Bridge, 22 water depth. It's an amazing seep site. When we flew over last night, there was a pretty significant gas plume going into the water column. Actually, there were 2. One is where we're landing, so that's going to be exciting. We'll just see gas when we hit the bottom. This site has gas hydrate, lots of animals, mussels, clams, all kinds of other fauna. But it's a really interesting site. I'm super excited.
There's tremendous interest in sampling the sediments. So for that, We have something called a push core or a tube core. It's basically a piece of acrylic in the shape of a tube, and it gets pushed down about a couple inches into the sediment. We retrieve it, and the sediment stays intact, and then the sediment is sectioned to look at what gases are present in the sediment, what bacterial species are present, if there are any larger organisms, any other associated invertebrates that are present, basically assessing the community of that sediment.
We're going down to the Pea Island seep field to look for earth burps, methane plumes at the bottom of the seafloor, and bacterial mats.
We're trying to figure out how the food webs connect, and so if we find the animals, we can actually run tests and find out, are they benefiting from any of the energy that comes from these seeps, or are they eating each other, or are they getting food from the surface? It helps us connect the dots to where the energy flow goes. Best scenario is we come back with an absolutely loaded basket. Lots of diversity. And I gotta go.
So I think one of the most interesting things about this project is the discovery and exploration aspect. This is fairly unexplored, and the images we saw today of these scarps, they blow the mind. Certainly wasn't expecting to see the diversity of corals on these walls that we're seeing here. So these environments along the East Coast are very dynamic. Look at the geologic map that Jason has up on his computer, and you can just see this giant divot that's been carved into the carbonate by the flow of the Gulf Stream. And these animals are living right on the side of that. And they're like holding on to the side of the canyon and getting the current blown by them, but that current is full of nutrition. There's a lot of talk about stagnation of currents with climate change, and I'm not saying it's going to kill the corals tomorrow. If that current slows down or becomes more stagnant or moves, then those corals lose their food source, right?
There are some corals in the Gulf of Mexico that were dated to up to 2,000 years old. So basically they're continuously growing little by little over years and years and years. These corals that we're working on are extremely slow growing and long lived. So fishing gear in particular, if you knock over a coral, that's less likely to be able to survive.
Having the opportunity to find these kinds of habitats and sample them here allows us to go to a big global map and say, okay, where else would these sorts of habitats be?
By understanding the places that we're working in now, that gives us a chance to model and predict areas that may have deep sea corals and seep environments. Ultimately, the data we're collecting will enable us to help with management considerations for other areas.
Yeah.
Subsequent dives are exploring coral sites where corals may be present on the sea floor.
I think we've really accomplished a lot in a short period of time. We've been able to do a dive within a canyon environment. We had an incredible dive at a seep, and now we're investigating a coral habitat.
After a dive to a vast Lophelia reef, Dr. Cordes is waiting for the results from a second dive to the same target at Richardson Hills.
We know what's way in the south and we know what's way in the north, and that's kind of in between. And so it would help fill it in, and it would increase my confidence that we're looking at like a 15-mile stretch of Lophelia reef. It's pretty crazy. Unknown until earlier this year. New discovery.
On August 23, 2018, the deep search team dove on Stetson Banks, 160 miles off the coast of South Carolina. Cheryl Morrison of the USGS was on board the Alvin to witness a surprising density of coral species.
We thought that was going to be just like an escarpment, basically rock. And so it was really surprising when we got there, and it looked very similar to what you all saw. Just coral along the edges and live coral on top.
What we've added up so far is, 86 miles, roughly?
We picked up on live corals pretty quickly, and the top was absolutely amazing, just live as far as you could see. You turn one way, all live corals, you turn the other way down the ridge, all live coral.
So now that we know this is here, the possibilities for where Lophelia could be forming reefs are now bigger than they used to be. And that's really the ultimate goal of deep search, is to be able to explore, find these things, inform the management agencies about the importance of this habitat, and then they can protect them. Limit as much as possible the human impacts in what are still relatively pristine coral reefs. You know, there aren't many of those left on Earth, and we just found a really big one.
The coral reef, eventually named the Richardson Reef Complex, turned out to be a national media story that provided valuable outreach to NOAA and BOEM, as well as priceless science to the public. The CTD rosette is an indispensable tool for gathering data in the deep sea. The CTD rosette measures properties in seawater, capturing water samples for further analysis in labs.
All of the environmental data that we get comes from a CTD, and we are seeing crazy patterns with how temperature varies in these areas.
Yeah.
The temperature profile is really weird here. All those waters are mixed. Everything that's happening up at the surface is very quickly being translated down to 700 meters. We see swordfish swimming all over them. You know, there's these big fish coming down to interact with these habitats. Just like you would, you know, build up a sweat trying to run a couple miles in 85, 90 degree heat. That happens to a fish too. And so they're down there in the deep water cooling down.
This entire large marine ecosystem is influenced by the Gulf Stream. We are seeing that in every data set. It looks like the corals are experiencing brief periods of elevated temperatures, and when that happens, the oxygen concentration temporarily decreases. So they're facing this double stressor of elevated temperature and reduced oxygen. We don't know right now if that's actually a stressful condition for them, because it's likely that they're either acclimatized or adapted to environmental challenges. That's something that we'll find out from the project. And I think that everybody's in agreement that thus far the cruise has been an overwhelming success. The discovery of the coral mounds at Richardson Ridge is really the pinnacle thus far.
Yeah, we're doing really well with collections, especially live corals. We have buckets of live corals to take home with us, which will allow us to really start to understand how they're able to live in this particular area. Because now, coming out here, we've collected the environmental data, so then we can simulate the environmental data back at the lab. We're really putting the pieces together during this cruise, and then in the next one that we'll have in April will give us even more data. What's nice about an ROV, and again, being able to use Jason in April, is that we will be diving for 24 hours a day.
The NOAA ship Ronald H. Brown, here departing from its home port in Charleston, South Carolina, in April 2019. This time, the Jason is on board to perform further exploration. The Jason is a remotely operated vehicle tethered to the Ronald H. Brown during dives. The ROV is able to send high-resolution video back to the control van where pilots control the vehicle, taking samples and navigating dive sites.
The cruise is kind of broken into two legs. The first leg will be working on the more southern habitats where there are large Lophelia mounds. The second leg will be going further north to the canyons and the seeps. Some of the things that we're looking at are coral diversity, biogeochemistry. We're looking at the fish and important fishery species and how the seeps interact with the surrounding environment through the microbes.
And USGS is primarily responsible for providing that scientific information to our partners.
That's what's so exciting, is that every time you put anything in the water, you're learning something new.
And it's about collecting baseline information. What is the geology and the bathymetry that leads you to find a deep sea coral reef stretching for 85 miles? If we know more about that system, then maybe we could predict where other sensitive habitats might be found in order to protect them.
And the other thing that's worth mentioning is at night, the survey team is conducting multi-beam surveys and mapping all of these areas so that when we come back the next time, we have really fine scale resolution for what the seafloor looks like in this area, which is something that didn't really exists before we came out.
Mapping efforts continue during all the deep search cruises.
You know, it starts with identifying where we need more data. You can put a box around something on a map and say, this is what we're interested in. This is where we think there's critical, habitats. We're collecting individual soundings of the seafloor, so individual depths. Each time we send out an acoustic sort of ping or sounding, we get about 800 soundings back. Over time, we get enough soundings to build up a picture of the seafloor, and then we actually physically make the maps. So we record where the seafloor is, and we record what's in the water, especially in these areas, the seep area like we're at now, where often you can see the plumes of the methane in the water. Certainly BOEM and NOAA and the USGS and everyone uses these results, and that really ties into the management side of things. This image is actually hopefully where tomorrow's dive will be.
Before this, we were blundering around in the dark. And it's really been two things. Multi-beam bathymetry, which shows us what the seafloor looks like, that can guide our expensive vehicles and the vehicles themselves. So what we have in this tank, there's 3 different species: Anolophthalmia profunda, Madrepora oculata, and then the dominant white species, which is Lophelia pertusa. That's the one that makes the reefs down here. The ones that were sitting on the little PVC fittings, they were attached to a cement block last year, and during the cruise, they were put back on the reef, recovered this year, and I'm going to look at growth rates and form using those little fragments that spent 9 months on the reef. I first got the idea of transplantation when I was working in the Gulf of Mexico on Lophelia, and there was vast expanses of seafloor that looked like it was perfectly adequate habitat, but there was nothing growing there. This experiment is useful because it shows us how Lophelia grows in this anomalous environment and how the other species grow compared to Lophelia. So this is an example of how we can maybe answer some of our questions using in situ experimentation.
This cruise on board the Ronald H. Brown has been a massive effort. At the end of the cruise, Dr. Amanda Demopoulos reviews the Deep Search team's achievements.
I was really impressed by the incredible imagery that we collected at the Blake Ridge Seep of a methane hydrate environment. It was an enormous mound with sediment covering the top, and then in this little cave area you could see this ice, methane ice, that had formed and created the mound, and then within the cave there were these fish hanging out, and it was the most enormous methane hydrate that I'd ever seen in my life. We set out to investigate some very extreme environments: canyons, chemosynthetic habitats, and deep sea corals. At the end of this mission, out here on the Ron Brown, using the JSON ROV, we have amounted at least 1,500 samples so far. We've been able to amass a tremendous amount of information in terms of the biological environment, who's living there, how diverse these systems are, and also the geological and chemical environment. And that information will then be used by the Bureau of Ocean Energy Management to be better stewards of the environment.
The Nancy Foster at sea in October 2019. The Deep Search team is on board, conducting the final cruise in the series.
On this particular study, I've actually been involved since the inception about 5 years ago, when we realized that there was a need for more information in this particular geographic area. Being here with the scientists helps us as managers to really focus more on what specific questions might need answering to inform our management needs. BOEM's interest is the very sensitive deep water coral that form three-dimensional structures. That can be incredibly important for fish species and invertebrates. And they sort of serve as kind of like a keystone species in terrestrial habitats where a lot of other species will depend on them or use them. So we need to know where they are well in advance of any leasing activities so that we can understand and describe what the impact to them would be should those activities ever take place.
Dr. Tracy Sutton, one of the principal investigators on Deep Search, is a professor at Florida Nova Southeastern University. Here, Dr. Sutton is conducting trawls, day and night. Dr. Sutton and his crew drag a finely meshed bag through the water column collecting species at various depths.
What we're finding is that the bottom communities and the water column communities are intimately linked together. We know from previous studies that this eats that. So we can start to put these things together ultimately for a model, an ecosystem model. That's what we would really like, because a model will allow you to make predictive hypotheses. If you affect this, that will be the trickle-down effect there. So these are the kind of interactions that we're looking for to build an ecosystem model that we can then form hypotheses and then go out and test those hypotheses. So identifying these habitats is absolutely critical. These communities in the open ocean generally form layers of dense life. Our goal is to first find out where these layers are. So for that we use the acoustics on board. So that gives us a large-scale synoptic view of the distribution of pelagic life, where you can see this deep scattering layer either collide with or pass over the top of these really dramatic features on the bottom, and seeing the intersection of those two, that's where a lot of life is happening. A lot of very interesting ecology is happening at those spots. Then we use midwater nets to sample those layers to find out what is in those layers that would then be forming the food source for those benthic communities. So by ground truthing, we're actually providing a quantitative estimate of life at the species level of observations that are made with different modes.
Dr. Furuminis of the Royal Netherlands Institute for Sea Research, or NIAS, is also working on board the NOAA ship Nancy Foster, tracking her lander with the use of a sophisticated USBL transponder deployed back in 2018. After sending a signal to the lander telling it to surface, the NOAA crew and the deep search team on board the Nancy Foster searched the horizon for a surfacing lander.
Being part of Deep Search is an amazing opportunity to actually monitor conditions over a longer time scale. This lander is like a platform, so we can mount it with lots of different instruments, like current sensors, temperature sensors, but also, for instance, cameras. We can measure what the environmental conditions are near the seabed for several months, up to even a year. And we use these data to actually identify why ecosystems in the deep sea are living where they live. So I think there's a lot to still be discovered.
The overall goals of Deep Search have been to explore the coral, canyon, and seep environments of the deep mid-Atlantic and provide the scientific grounding for their management. The Deep Search team made 25 dives in submersibles, ROVs, and autonomous vehicles during 6 separate expeditions. 940 hours of video and 4,004 individual samples were collected and cataloged, including 11,280 liters of water. New advances in the use of sound were used to monitor deep-sea communities. Invaluable data were also gained by collaborative work with the Okeanos Explorer and ADION programs. These studies have improved our understanding of the role of methane seeps in providing habitat and food to the deep sea, the importance of the canyons in supplying nutrients to shallow waters, and the interactions between the fish communities of the Gulf Stream and the deep reefs. All of these efforts help categorize the tremendous biodiversity of species in the ocean and the changes taking place in these vital habitats.
I think this project is going to contribute a lot to the future of this whole area.
We have the samples, we have coral, but this is step one. All of that's going back to, 9 different labs to be looked at? So there's a lot that stands to be learned.
We can't do it alone. We have a suite of academic partners, and by bringing all of these different scientific experts together, we can improve the information gathered for every federal dollar spent offshore.
This kind of research is really challenging, but I think that we collected a lot of really good data. We know more, for example, about where Lophelia occurs than we did just a year ago. But to actually go out and see the habitats that you're trying to protect, that's a pretty powerful thing.